So I'm going to talk to you today about photosynthesis, Earth system models, and the Arctic. A little bit of background about how I became a scientist at BNL. So throughout the talk, I'll try and sprinkle a little bit of that in, and hopefully that'll help you understand how I made that postdoc scientist transition. So photosynthesis is the largest single flux of carbon from the atmosphere into the terrestrial biosphere or into any other part of the planet. About 120 gigatons of CO2 comes out of the atmosphere every single year, is drawn down by photosynthesis, and it enters the terrestrial biosphere. And then, roughly balanced, these two processes of autotrophic and heterotrophic respiration return that CO2 to the environment, to the atmosphere. Has that mic gone out again? Maybe I'll have to just stick with this one. So photosynthesis, at the heart of photosynthesis, there's the enzyme Rubisco. This is in the dark reactions of the Calvin cycle. And this enzyme is responsible for taking ribulose 1,5-bisphosphate, a 5-carbon compound, adding a CO2 to it, and producing 2 3-carbon compounds. The energy for this process comes from the light reactions, where sunlight is harvested in the thylakoid membrane, and energy in the form of ATP and NADPH is used to power that Calvin cycle. And the product of these two processes is the sugar and starch. So Rubisco evolved about 3.5 billion years ago. I'm not sure the exact day, but what we do know is that the CO2 concentration was really, really high at this time. And through billions of years of geological processes and eventually the evolution of photosynthesis, the CO2 concentration was drawn down. And as land plants started to appear, the atmosphere started to get polluted with oxygen. The problem with this is that Rubisco also, it turns out, has a reaction with oxygen, the oxygenase reaction. And in this reaction, this 5-carbon compound reacts with oxygen to form a 3-carbon compound, which can go back into the Calvin cycle, and a 2-carbon compound, 2-phosphoglycolate, which is poisonous, and the plant has to try and get rid of this. And they evolved a pathway called photorespiration, which returns a 3-carbon compound to the Calvin cycle and salvages about 75% of the carbon. The downside of this is that this process also releases some of the CO2 that was fixed by photosynthesis. So at current atmospheric conditions of CO2 and a temperature of 25 degrees, for about every 3 to 4 carboxylation reactions which are fixing carbon, we're losing carbon through this oxygenation reaction. The enzyme also has absolutely miserable catalytic activity. It's one of the slowest enzymes on the planet. If you compare it to PEP carboxylase, which also fixes CO2, you can see the difference in the turnover number of the enzyme there, which is the number of reactions it can process per second. So if you think about it, this enzyme's got an oxygenase reaction, which has a negative impact on its primary function, and it's also incredibly slow. It happens to be the most abundant enzyme on the planet, so it's not exactly the best poster child for intelligent design. So kind of to give you a little clue about how I got into science, I'm just going to try and take you through some of the steps. So back when I was starting my BSc, CO2 concentration is about 356 parts per million, and the IPCC had just put out the first climate change report. And I started at this time, my degree was originally in animal physiology, and I switched over to plants pretty quickly. And one of the reasons was that I started to draw the link between climate change and plants and understanding the central role that plants have in determining the rate of global change. So, current numbers suggest that we're putting about 10 petagrams or 10 gigatons of carbon into the atmosphere every year in terms of fossil carbon fuel emissions. And these have risen steadily since the beginning of the Industrial Revolution. This has translated into a rise in the atmospheric carbon dioxide concentration as measured by the classic data set of Dave Keeling, which started in the late 60s, and also by popping bubbles in ice cores, which allow you to go back to the start of the Industrial Revolution and much further and measure the CO2 concentration in gas trapped in ice cores. But not all of the CO2 that we burn ends up in the atmosphere. So here's our 10 gigatons from fossil fuel, about another gigaton from changing land use, and about half of it ends up in the atmosphere, and the rest is absorbed by the land and the oceans. I'm not going to talk about the oceans, but basically it's mostly a chemical and physical process which drives that, and it's, in comparison to the terrestrial biosphere, is very well understood, where the response is mostly biological and therefore a lot more complex. So the reason that the terrestrial biosphere can absorb this additional CO2 is that the carboxylation rate of this central enzyme, rubisco, is stimulated when you have additional CO2. This reaction, the carboxylation reaction, is limited by the availability of the substrate. And in addition, when you do increase the availability of this substrate, you competitively inhibit these oxygenation reactions. In addition to the rise in temperature, or because of the rise in CO2 concentration, we've also now got an unequivocal rise in the temperature. And this graph shows the temperature anomaly, and it's about a 0.75 to 1 degree rise since the beginning of the Industrial Revolution. And temperature also affects photosynthesis and the response to rising CO2. Shown here is a wheat leaf that I've modeled CO2 assimilation rate for based on 1990 CO2 levels. And you can see that as the temperature increases up to an optima, photosynthesis increases and then it decreases. So if you're a wheat plant growing at 25 degrees, global warming is not going to help your photosynthetic rate, but if you're trying to grow wheat in Canada, it might be quite beneficial. When you add in CO2, you can see that the effect's much greater in terms of stimulation. So this is the same leaf modeled at CO2 concentration that you'd expect at 2050. And what you see here is there's a couple of things. Firstly, the temperature optima is shifted as you go to high CO2, and also the stimulation at low temperatures is much lower than at high temperatures, and that's because high temperatures favor the oxygenation reaction of rubisco, and the impact of elevated CO2 is therefore much greater. So as I was finishing up my BSc and starting my PhD, Brookhaven Lab had just developed this technology called free air CO2 enrichment. And this was really enabled scientists in the field to answer difficult questions that they've been trying to get their hands around for some time, which was what happens to plants in their natural environment. So one of the problems with conducting experiments in controlled environments was that the chambers that you grow the plants in and the pots that you grow them in often cause large artifacts in the interpretation of the data, and there was a real need to see what happened in a natural environment. And Keith Lewin and George Hendry developed this free air CO2 enrichment technology. And the way this worked was that you had a measurement of wind speed and direction, a sampling of the CO2 in the center of these plots, and then the upwind vent pipes would release CO2 across the vegetation, and therefore you could understand what was happening to plants basically in their natural ecosystem, except all you've done is fast forward the CO2 concentration by about 50 years. So as I finished my PhD and I was looking for postdocs, Brookhaven seemed like a good place to come because it enabled me to have the opportunity to go and work in some of these sites. This included this face site in North Carolina, which was a loblolly pine plantation. You can kind of get a sense of the scale there. This is a person standing on a central tower. Also, there was other sites. There was the Aspen face site in northern Wisconsin, a soybean field in Illinois. And all these experiments gave me opportunity to go and conduct work in these different ecosystems. And one of the unifying things we were able to do is that use models to try and understand the responses that we were observing in the field. And with photosynthesis, we're quite lucky compared to other areas of ecology in that there's a really good model that was put out by Farquhar, von Kamer, and Berry in the 80s. And this describes photosynthesis, if it's parameterized well, it describes photosynthesis really quite well. And basically, photosynthesis is limited by two major processes, this WC and WJ term. And I'll just explain those a little bit. So the WC term refers to the dark reactions of photosynthesis, which I talked about earlier. And the WJ refers to the light reactions. And the biochemists might notice the Michaelis-Menten equation here, but basically the main driver of this rate is this term VC max, which is the maximum carboxylation rate of the enzyme Rubisco. And then the main driver of the light reactions is this term J, which is the electron transport rate. And you can measure these parameters in what's known as an ACI curve. You can hold the leaf level, relative humidity and temperature constant, hold the light level at saturating light, and then you can vary the CO2 concentration that the leaf's receiving, and you can estimate this response curve, which would start here at low CO2, where VC max is the limiting process, and then as you reach this inflection point, now the second process, Jmax, becomes limiting. And this is key to understanding the CO2 responsiveness of plants and their ability to absorb CO2. So this sort of, going back to kind of the career thing, I think, like in my postdoc, this was a critical moment here. So in the midst of going around these different phase sites and starting up experiments, I took some time to work with a colleague of mine, and one of the things I ask people when I interview them for positions is I say, what paper are you most proud of and why in your publication record? And this was mine, and this was the first paper where I conceived the idea, and with some help from Steve, executed it, and then published it and got it into a good journal. And one of the challenges for postdocs, I think, is that your scientific record is assessed not really on the impact of the papers, but it's assessed on the impact of where they are. So, Global Change Biology is a pretty good journal in our field. So, I got the paper in there, and that was great. And my managers could see that I had the ability to work independently and deliver on a publication in a quality journal. I mean, they're not to know that this paper was going to go on and get 50 plus citations at the time they have to make a decision. They're making it just on the title of the journal. And I'd say that one of the critical things I think for postdocs is to publish, and to publish in good journals. So what we found in this study was that when you grow plants at elevated CO2, often the initial stimulation you see in photosynthesis isn't maintained over time. It diminishes, and this is known as photosynthetic acclimation. And we were trying to work out which process might be determining that. And this figure here shows the ratio of photosynthesis at elevated CO2 to current CO2, and this would show the initial or the theoretical maximum stimulation at elevated CO2 in this kind of summary or meta-analysis of different studies. But the actual photosynthetic rate that was observed in these studies was significantly lower. We used the Farquhar model to work out that this decrease in photosynthesis was due to a reduction in VC max. So then, with a couple of good papers under my belt and a few in the pipeline, there was an opening at B&L. As one guy had just left to start a job somewhere else, and I was fortunate enough to be in the right place at the right time with a decent enough record where they were able to hire me. And I remember saying to my manager at the time, I saw him in the corridor after I'd found out, and I said, thanks for taking a chance. And he looked me square in the eyes and said, oh no, I didn't take a chance. And to me, that was one of the nicest things anyone's ever said to me, and it was a really, really great vote of confidence. And I think a lot of that is that those early publications gave him the ability to make that case that I was going to be worth gambling on, or in his case, not gambling. So what I want to do now is fast forward to 400 ppm and talk about a new project which we've been working on, which is called NG, which stands for Next Generation Ecosystem Experiments. So the NG experiments, the idea behind this is to improve the representation of critical processes in Earth system models. And the motivation for this is basically that the models aren't doing a very good job. So since we had this first IPCC report in 1990, we've been learning more and more about the causes and attribution of global change. But yet our ability to model the future is still not that great. We know it's going to be bad, but we don't know how bad it's going to be. And this is of interest to the Department of Energy because they want to know how bad it's going to be. And one of the key things that they have to get right is the terrestrial biosphere. And here's an example of the problem. So this figure shows the annual flux in petagrams per year of carbon into the land. So this is a measure of the terrestrial carbon sink. This is the carbon sink that's basically subsidizing our fossil fuel use. So if this sink disappears, that's a big deal for global change and the rate of global change. And these lines, these are all Earth system models. These are the ones that the IPCC uses to tell us what's going to happen in the future. And what you can see here is that there's a large disagreement amongst these models about whether the land is going to be a source for carbon in 2100 or a sink for carbon in 2100. So there's a great degree of uncertainty. And this was a model into comparison project. And this was done in 2006 and preceded the 2007 IPCC report. And then we had the latest IPCC report, which was preceded by, they call it CMIP5 this time. And you can see that basically we haven't got any better. And there's still this major problem with working out what's going on with this terrestrial carbon sink. And so that brings us back to the motivation for DOE and these ENGIE experiments, is that they're trying to focus on systems which are climatically sensitive, globally important, and understudied or poorly represented in the models. Right now, we're working on putting a proposal together for a project called ENGIE Tropics, which, not surprisingly enough, is going to be focused on work in the tropics. But what I want to talk to you today about is ENGIE Arctic, which has been running for 2 or 3 years. So the Arctic is climatically sensitive. The warming in the Arctic has been much greater than it has been over the planet as an average. After a long cooling period, at the start of the Industrial Revolution, we've seen about 1.5 degrees of warming in the Arctic. And this has real impacts, and you often hear about ice retreats. And a couple of years ago, I was in Barrow, and they had a particularly warm year when the Northwest Passage opened up to commercial vehicles, commercial vessels. And we were in Barrow, and a cruise ship from Scandinavia had come through the Northwest Passage. It was the first time that had happened. And they launched their boats and were putting all their tourists aboard on shore. And they're all just like beached these boats. I mean, there's no infrastructure in Barrow at all. And climbed out of the boats in their red jackets and were wandering around town. And people were like, well, is this really a port of entry? And how are we going to do this? And the people up there hadn't really experienced tourists arriving from Europe, without passing through a U.S. port of entry, and they were kind of not sure what to do about it. And that to me was like, wow, that's a real example of climate change that you're seeing firsthand, that you see these kind of things. Alaska is going to continue to warm rapidly, as is the rest of the Arctic. Here's a couple of emission scenarios. This is from the National Climate Assessment, so of course it's in Fahrenheit, not Celsius, so I apologize for that. But basically, the message is pretty clear that for Alaska, you're looking at some incredibly large increases in potential temperature by the end of the century. This is of a major concern because Alaska contains, or the Arctic contains, 50% of the global organic carbon reserves. So this is frozen organic matter, which is currently frozen in permafrost. And what's happening is that as the temperature rises, the active layer of the permafrost is deepening, and then it's exposing more of this carbon, which was previously not part of the carbon cycle, or hadn't been for millions of years, to microbes that degrade it and release methane or CO2 into the atmosphere. And this is a really important potential feed forward on climate change as it gets warmer. The permafrost thaws and degrades a bit more CO2 gets released, which makes it hotter, which then feeds forward on the warming and release of CO2. So this is a critical thing to try and understand and try and get right. At the same time, permafrost thaw and degradation also releases nutrients for plants and allows a deeper rooting volume for plants. And the effect of that is that you see this greening of the Arctic, which is where plants are kind of moving northwards and colonizing areas which were previously difficult to colonize. So what we're seeing there is this potential trade-off between, will the Arctic allow a greater gross primary productivity from increased plant life, or will that be offset by CO2 release from previously frozen organic matter? So we were tasked with trying to solve this problem, and it's a DOE project, so of course we have a deliverable, which is this ecosystem process model, a highly process-rich model that really allows scientists to try and understand what's going to happen to the Arctic. Right now, a lot of the key processes aren't even represented in the model, and my part of this was understanding photosynthesis. So as a first step, we went to have a look at what are the major model sensitivities. And this figure shows a model uncertainty analysis. So what they do is they take 80 parameters from, this is the community land model, which is the landmark US model. They take 80 parameters from this model and vary them all and find out what the impact on various model outputs are. And you can see these model outputs here. So this is total vegetative carbon, leaf area index, heterotrophic respiration, GPP. And what you'll notice is these darker bands that have the greatest impact on model output are actually connected with photosynthesis. So this FLNR, I'll talk about this later, but this is the fraction of nitrogen that a leaf decides to invest in the enzyme Rubisco. And then this leaf CN ratio basically is used to compute leaf nitrogen content. So the next thing I did was I then went out and looked at all of these different Earth system models and the component models which feed into them, and to try and understand how they parameterize this important parameter VC max. And these are the acronyms for all of these different models. And these are the ones that are used to drive the models, the Earth system models that are in the IPCC report that tell us what the CO2 concentration is going to be in 2050 and 2100. And you can see that there's just a huge variation in the values. These are plant functional types. So this, for example, would be needle-leafed trees growing in the temperate region, broadleaf deciduous trees, boreal. So the way these models work is that they carve the planet into different groups. Some models will use 5 plant functional types to describe the entire planet. Some of the more sophisticated ones might use up to 16. So what I did was I pulled them all together and then assigned them to different plant functional types, just to see what the spread was. And it's enormous. Even for like broadleaf deciduous trees in the temperate region, which grow outside most major universities, there's still a massive disagreement about what we should be using as a VC max. And the dirty secret of the modelers is that this parameter is so important that they actually use it to tune their models. I heard an anecdote at a workshop recently where the UK model, some of the surface runoff guys were using VC max to tune surface runoff in their global models, which is just mind-blowing. So as part of the project, we're kind of focused on the US model, the community land model. And so I wanted to have a look at how they represent VC max and how they determine it. And this is the equation they use. So we've got VC max equals leaf area multiplied by this fraction of leaf nitrogen invested in Rubisco, multiplied by this other number, which is how much nitrogen is actually in the Rubisco protein itself, and then multiplied by a constant, alpha R25, which is the specific activity of Rubisco. These two are assumed in the model to be global constants, and these two would be assumed to vary by plant functional type. So I had a look at, sort of dug deep into their papers and tried to work out what was going on. And then I found my first mistake. So what they'd done to estimate this term FNR, so this is simply the amount of nitrogen that's in the protein Rubisco, and it shouldn't really be any different from any other protein. Maybe there's a few quirks here and there, but basically it should be the same. And what they'd done was they'd got this number 13.96, and this just made me sweat when I first saw this, because I'd been using 16 in my papers for the last 5 years, and was like, oh my God, have I made this terrible mistake? Because these are the Earth system modelers, and they should have this stuff right. So I went and looked at their proximal references, and there's this paper from the 50s or 60s, I can't remember which, where they'd done this nice work where they'd got the protein, this is pre-sequencing, and they'd chopped it up into its constituent amino acids and provided a table of all the different amino acids. And what the guys who built this model had done was said, okay, great, so they added up all the amino acids, worked out how much nitrogen was in them, and then came up with this value of the amount of nitrogen in the enzyme Rubisco is 13.96%. And I looked at this and I was like, but they haven't joined these amino acids together to form a protein, so they'd omitted the fact that the amino acids are joined together with a peptide bond, which results in the release of a water molecule. And if you do that, you end up with a number of 16%, so I was able to relax a little bit about my previous papers. And then the other thing I did was I looked at this term specific activity. So you can kind of unpack this a little bit into its constituent parts. And CLM uses this a value of 60, and this was measured pre-sequencing, pre-sophisticated measurements, and they've just kept that value. But if you But we know various different things about this. So in Rumsfeldian terms, we know that kcat can vary, but the best data we have right now is that it's about 3.3 per second for C3 plants. C4s are about 4 per second. This model is also assuming that Rubisco is 100% active, which is a reasonable assumption, but we don't really know that for sure. And that assumes that there's 8 active sites available for carboxylation or oxygenation. And we also have a really solid idea about the mass of the enzyme. So at the end of the day, math says it's 47 and not 60. So now we've just got 2 parameters which I've just shown have been decreased by a considerable amount. And I asked one of my colleagues to run this through an Earth system model in an uncoupled way, which is a way of saving computer power. And this is a result of that. So what they found out was that basically, so those corrections are global, so the biggest impact is where you have the most primary productivity, which is in the tropics. And what this figure shows is the model run with their original calculations versus the model run with my corrections. And what you can see here is that there's a large difference in the amount of carbon that's being fixed by the planet. And if you put that in terms of gigatons per year, that's 10 gigaton per year error, or 10 petagram per year error. So that's the same magnitude as our global fossil fuel emissions. That's an error in the model in the models that were used to produce the IPCC report that's sitting on your desk at the moment. So then, because we're focused in the Arctic, we wanted to go and have a look at what's happening to these other 2 parameters, the leaf nitrogen content, the fraction of leaf nitrogen in Rubisco, and also because we wanted to measure VC max as well. So you can't do this in your office, you have to get on a plane. So I fly from Long Island up to Barrow, 16-hour plane ride, multiple switches, there's no roads, it's a difficult place to get to. But there's a rich history of science there, and there's some pretty cool logistical support. And this is what it looks like up there. You have this polygonal ground, which is formed by ice wedges, and it's a really kind of uniform-looking topography. Very, very flat, very low stature vegetation. The growth season for these plants is about 53 days a year, so for the past 3 or 4 summers, I've been wearing thermal underwear and missing out on Long Island summers and switching them up for Arctic summers. I'm kind of getting tired of it now. And we go out, the place where we work is the Barrow Environmental Observatory. This is a protected area. The local Inupiat population recognized the value of science in their community, and they protected a huge area of land and dedicated it for scientists. And this is really important because there's a lot of pressure on this land right now. There's a lot of oil activity. They were trying to drill in the Chukchi and Beaufort Sea when I was up there one year, but they failed to do that. They had some problems with ice, so some of the locals were happy about that, some of them weren't. But the community really supports science, and so this is a great resource to be able to do work in this area. And what we tried to do was to go and measure plants that represent different plant functional types. So we've got global plant functional types, but there's also plant functional types for different ecosystems. So we tried to understand what was going on in these different PFTs. And the way we did this was we went up and we measured ACI curves, which I talked about earlier, and then also collected samples for leaf nitrogen and other analysis. And the way you do this is with an infrared gas analyzer. So this is the control unit, and this is the business end. And you basically clamp this on a leaf, and you can control temperature and light and relative humidity. You can get those constant. You wait for the leaf to stabilize, and then you conduct your experiment, which is you change the concentration of CO2 that the leaf's seeing, and that enables you to build this ACI curve. And then from that, you can estimate these 2 key parameters I've been talking about, VC max and J max. So we get up to Alaska, and then we start to realize why there's not much data in that part of the world, and there's not much information to parameterize these models. With the gloves we're wearing there aren't to protect the samples, they're to prevent the styluses of mosquitoes getting through to our hands. This is an incredibly difficult place to work. The instruments themselves give out CO2, so it just brings the mosquitoes in. So you're sitting in water surrounded by mosquitoes. And I have a little video here which just kind of gives you an idea of what it's like. These things get absolutely everywhere. They get in the optical path of the instrument. You have to have two people to open and close the chamber to get the leaf in, because otherwise you find yourself measuring insect respiration instead of photosynthesis. And the amount of effort it takes to collect these data, I mean, these are some extremely hard-fought data. They also get inside the instrument control panels, so you can't see what's going on the control panel. And they also jam up the cooling fans. I was doing an experiment one day, and the instrument just started failing. I was like, oh, what's happened? Has the leaf crashed or something? And then a few days later, when I had some time to pull the instrument apart, I found out that the reason was that the mosquitoes had been sucked into the air intake of the analyzer and had literally clogged up the cooling fan with their dead carcasses. And it's just, I mean, these are just the kind of things that make it really difficult to work up there. So we got bug suits for the instruments now as well, so we don't have that problem. So here's the data we got. So this is VC max measured in a number of these species which represent key plant functional types. And this here is the representation of VC max in some of these Earth models. Only 4 of the Earth models explicitly include representation of Arctic plants. And you can see here that all of them are underestimating the photosynthetic capacity in the Arctic, and by quite a large degree. The other thing that we did as well is that we've done temperature response curves, so we've measured VC max at different temperatures. One of the things that's really important to get right in these models is this temperature response. And typically, some of the models are actually using temperature responses which are so outdated, and we've learned so much more that they're fundamentally flawed, but they're not allowed to update them until the hydrologists and all the other components of the models agree, because it might crash their model. So it's a very slow process to get progress in these models, and this shows the US model, which is using one of these modern temperature response curves. And what we see here is that our data are not tracking this. And if you were to take values of VC max and try and scale these with temperature, you would basically underestimate the capacity at low temperature, and that's what we saw in our other data as well. So the next question I asked was like, okay, we've worked out these two terms, we've got a measure for this, we can measure these two. Let's find out why CLM's underestimating VC max. So we measured leaf nitrogen content, and here's the value for CLM. So it's on target for some of these species actually, but then it's about half for some of the other ones. So that, on average, they're underestimating leaf nitrogen. But we also went and looked at this term FLNR. This was this term that in that sensitivity analysis I showed you was one of the glowing red parameters. And this dashed line here shows the levels that CLM uses. And we used this equation and solved for FLNR. And those are shown in the circles there. And you can see that our estimates of the investment in Rubisco is much, much higher than CLM. And then we also did an independent biochemical method using C14 labeling of an inhibitor which binds the binding sites of Rubisco. I had some colleagues at University of New Brunswick help me with that. And in some cases we agreed, and in some cases they were different. This work's ongoing. We're going to try and work out what's going on there. But in all cases, these values were much, much higher than CLM. And anybody who's ever run an SDS page gel of a plant leaf will know there are some pretty enormous bands there in their Rubisco. And the assumption that CLM's making in some cases for some plant functional types are that the investment in Rubisco is like 4 or 5%, and that's just simply not true. So basically what they've been doing is having an uncertainty over this value is being used to tune this one. Sometimes they're just tuning this one directly so that their model outputs match current day observations from flux towers or remote sensing. So what we're hoping to do with this data is to try and constrain this part of the model so that they can't mess with it, and they have to go and find something else to tune with, or to go and work on another process or identify a process that really needs a lot more work, as does photosynthesis. So just to summarize this part, so these are global constants. We found that CLM was overestimating both of these. So this resulted in the model, and the model had a history of having an overproductive tropics. So this in part may explain that. But also, we had big changes in the leaf nitrogen content, and also the investment of leaf nitrogen in Rubisco. So in conclusion, plants subsidize our fossil fuel emissions. by taking CO2 out of the atmosphere and sequestering it in the terrestrial biosphere. It's about a third of the a third of the CO2 that comes out of your tailpipe is ending up in a terrestrial biosphere somewhere. The problem is that when we're trying to represent these processes in Earth system models, which can give us the confidence to make policy decisions based on, do you want to build a 16-foot levee or a 25-foot levee? These decisions are based on these models, and to get that right, you need to know the CO2 concentration in the atmosphere and what that temperature translates to and what that means in terms of sea level or food security. So making these decisions, the policymakers are trying to use data which is in no way commensurate with the level of confidence that we have in the projections right now. And I'd just like to talk a little bit about some of the next steps we're taking. Sean Serban, who joined our group in March, has been one of the pioneers of using spectral signatures to measure the properties of leaves and link those to physiological traits. And here, this is one of the figures from his paper, which shows that the observed VC max can be predicted from spectral signatures. So this is great news for me. It means I don't need to spend as much time getting bitten by mosquitoes, and we can use this very quick method to get good estimates of VC max once we've calibrated it for the systems we're working in. And the evidence that Sean's seeing so far is that his relationship's holding up in a number of different biomes, which is really, really encouraging. The cool thing about spectral signatures is that you can fly instruments on aeroplanes and potentially in the future use satellites which can give you the same data. So that enables you to make maps, and this is a map that Sean's made of an agricultural field in California. So this pixelation is not the data, this is actually different fields of different crops. So this enables us to provide temporally and spatially resolved trait maps of these really important traits for these models, and that's where we're hoping to go in the future. Right now, we're kind of at the leaf level, and in Barrow, we also have a 60-meter tram, which is running up and down, taking various different measurements of the ecosystem underneath it. We're starting to play around with UASs to mount some small instrumentation, also using leveraging overflights when we can. And ultimately, from space, there's a satellite that NASA's hoping to put up, or it's being talked about, called Hysperi, which would have a full spectrum hyperspectral camera looking down on the Earth, which for us is really exciting because it enables us to make a global map of these really important traits and then see how they change with the seasons. And this would be an incredible resource for the modelers, because now they've just got the data that they can use to start running their system. And with that, I'd just like to say, acknowledge the support of the Department of Energy and say thanks to my colleagues at NG Arctic. There's tons of really cool pictures. It's an amazing place to go, and you see some incredible things. So go and check some of these pictures out. They're a lot of fun. And also, if you're really excited, you can go and check out our website or follow us on Twitter. Thanks very much. Do you want to just use this one?
Okay, thank you very much. We have a few minutes for questions. So if anybody, I guess.
Yeah, I missed when you corrected those parameters. Was the impact that the plants would be able to absorb more CO2?
Yes.
Or less? They'll be able to. So it's good news.
Yeah. So Paul's question was, what's the result of my corrections on those models? And the answer is that VC max would be increased, which would give the plants an increased capacity to absorb CO2, which is good news. That doesn't mean that we're suddenly going to get this like extra drawdown of CO2 because I've just written this paper. But what it does mean is that we understand that system a little bit better.
Do we have any more questions? Actually, I have a question more from a postdoc career point of view. So when you transitioned from a postdoc to assistant scientist or whatever, did you find anything surprising or difficult with it? Was there anything you didn't expect?
That's a good question. I think if you're ready to make that transition, it's not that unexpected. I mean, You know, I had a kind of a supervision issue when I was a postdoc, so I was working incredibly independently already, and I think that probably helped the managers have the confidence to hire me. So I didn't notice a big transition, or there wasn't a big step change culture shock. But of course, there's other things that you now have to deal with, like supervising people and dealing with budgets and the politics of science, which you would usually shielded from. So those things are difficult to handle. But in terms of just doing the science, I think it wasn't really a step change.
Thank you. Anybody else? Well, let's thank Dr. Rogers again. Thank you very much.